Are you ready for the OPRA exam? Welcome to Pharmalytics. I'm Ava Patel, and today we're putting your pharma knowledge to the test with a 10-question OPRA mock test. I'll present each question with multiple choice options, give you 10 seconds to answer, then reveal and explain the correct answer. Grab a pen or keep a mental tally. Let's see how you stack up. First up, which excipient acts as a binder in tablet formulation? A. Magnesium stearate. B. Starch. C. Talc. D. Aerosol. 10 seconds. The answer is B. Starch. Starch binds powders together, giving tablets their solid form. The others, magnesium stearate, lubricant, talc, glidant, and aerosol, flow promoter, have different roles, but only starch is the binder. Next, which sterilization method is suitable for heat labile solutions? A. Autoclaving. B. Dry heat. C. Filtration. D. Radiation. 10 seconds. The answer is C. Filtration. Filtration removes microbes without heat, perfect for sensitive drugs. Autoclaving and dry heat destroy heat labile drugs, while radiation can degrade some chemicals. For delicate solutions, filtration is the gold standard. Question 3. The half-life, T12, of a drug depends on which factors? A. Volume of distribution and clearance. B. Dose and absorption rate. C. Root and protein binding. D. Metabolism and excretion alone. 10 seconds. The answer is A. Volume of distribution and clearance. T1 to 2, 0.693x VD/cl. A larger volume or slower clearance means a longer half-life. Dose and absorption affect peak levels, not half-life. Which test checks tablet hardness? A. Friability. B. Disintegration. C. Hardness. D. Dissolution. 10 seconds. The answer is C. Hardness test. It measures the force needed to break a tablet. Friability tests resistance to abrasion. Disintegration checks how fast it breaks apart. And dissolution measures drug release. Only the hardness test measures crushing strength. Which organ is the primary site for drug metabolism? A. Kidneys. B. Lungs. C. Liver. D. Intestine. 10 seconds. The answer is C. Liver. The liver's enzymes, especially CYP450, handle most drug metabolism. Other organs help, but the liver is the main metabolic hub. It's the body's chemical processing center for drugs. What is the function of a buffer in a pharmaceutical solution? 10 seconds. The answer. Buffers maintain pH stability, protecting drugs from degradation and precipitation. They keep drugs dissolved and minimize irritation for injections or eye drops. Buffers ensure a stable, effective, and safe formulation. In chromatography, what does retention time depend on? 10 seconds. The answer. Retention time depends on how strongly the analyte interacts with the stationary phase. Stronger interaction means longer retention. Weaker means faster elution. This principle underlies all chromatographic separations. Which pharmacokinetic phase involves absorption and what happens during it? 10 seconds. Absorption is when a drug moves from its administration site into the bloodstream. It's the first step for most drugs, determining how much reaches systemic circulation. The bioavailability of an IV drug is 10 seconds. The answer, 100%. IV drugs go directly into the bloodstream. No barriers, no loss. This route provides the fastest, most complete drug delivery. All other routes are compared to IV for bioavailability. Which dosage form bypasses first-pass metabolism? 10 seconds. The answer. Sublingual tablets, and also buccal, transdermal, inhalers, injections, bypass the liver's first-pass effect. Drugs absorbed this way enter systemic circulation directly. This is crucial for drugs rapidly metabolized by the liver. That's the end of our OPRA mock test. How did you score? 0 to 4. Review the basics. 5 to 7. You're on track. Keep practicing. 8 to 10. Excellent. You're OPRA ready. Download our free OPRA study plan in the description to structure your prep. Subscribe for more mock tests and resources. Let's ace this exam together.